Thank you very much for the invitation, and thank you very much for all coming. I'm David Russell. I'm a postdoc in Claudia Bonfio's lab in ETH Strasbourg. Prior to that, I was a postdoc with John Sutherland in Cambridge, where I worked on all manner of aspects of prebiotic chemistry. Before I start, I'd just like to ask how many chemists are here in the audience. Can I see some hands, please? A fair number. And physicists, and geologists, and astronomers, and planetary scientists, and any other types of scientists that I haven't been able to identify. Excellent. You can fit in many categories. Okay, this is going to be a fairly chemistry-heavy talk, so good luck to you all. Let me start by putting to you a simple question. What do we mean by prebiotic chemistry? I will give a pretty simple definition, and that is the chemistry that could have taken place on the early Earth before the advent of life, and that could have led to the building blocks of life and thence to life itself. Let me follow up by asking a second open question, and that is, what is the aim of prebiotic chemistry? I would suggest that it is to delineate a path from inert chemical matter to something that we would all agree is alive. How far we are from achieving that goal, well, I'll leave that up to you. But I would say that while we may be the ash of burnt out stars, the nuclear waste of the cosmos, some remarkable chemistry got us here, and I think we can all agree on that. Okay, so today I'm going to tell you 6 short stories extracted from 6 papers that are seminal to the field of prebiotic chemistry. These papers date from the period 1953 to 1977. And this was a period in which the field of prebiotic chemistry was emerging, much like a planet might emerge from a protoplanetary disk, like we saw earlier. I would add that the questions raised by these studies are still very relevant to today. Not all of these questions have been resolved, and much remains to be discovered. Okay, so in each case, I will talk you through the principal experiments discussed in the paper. We can have a look at the results and their analyses, and we will discuss their significance. Okay. Where else could I start but with the now famous Miller-Urey experiment? How many of you have heard of the Miller-Urey experiment? Very good. How many of you have read the original paper? You're not on camera, so you can be honest. That's kind of what I expected. Okay, let me tell you about it. Well, the first thing to note is that there's only one author on this paper, and that is Stanley Miller. Harold Urey was the very model of a supportive doctoral supervisor, and he did not want to take any of the credit away from Stanley Miller, whose idea it was to experimentally test some earlier hypotheses as to the origin of life. And in fact, the story goes that Urey was instrumental in getting the journal Science to accept the paper, because after 6 weeks of silence, Urey wrote to the journal editors and said that if you don't publish this paper, we're going to retract it and send it to JACS. I can't imagine anybody doing that nowadays, but it worked. Okay. So let us begin. Miller writes that the idea that organic compounds served as the basis of life were formed when the Earth had an atmosphere of methane, ammonia, water, and hydrogen, instead of the carbon dioxide, nitrogen, and oxygen that we find in the atmosphere today. So the idea is that the prebiotic atmosphere was reducing, that there would have been some hydrogen content and potentially some ammonia around. He then goes on to say, to test this hypothesis, an apparatus was built to circulate the gases methane, ammonia, water, and hydrogen passed an electric discharge which served as a model for, say, lightning in the atmosphere of the primitive Earth. And they analysed the resulting mixture of compounds formed by paper chromatography. So there we are, a nice, simple way to experimentally test an hypothesis, and what a succinct explanation of the work involved. Let's now take a look at the apparatus. This is now quite a famous apparatus, but at the time it was bespoke and unique for this set of experiments. So this is an all-glass apparatus in which we have a flask down at the bottom in which some water was boiled. We have a long glass tube which connects to this essentially reactor space where the mixture of gases can freely circulate. This mixture of gases passes between two electrodes where the spark discharge occurs. There is a condenser which enables the mixture of gases and water vapor to condense down into this U-bend, and then when the volume of the U-bend is sufficient, it drains back into the main flask where it continues to circulate around. So this is how they did the experiment. They added water, Miller added water to the boiling flask, about 200 mils. He evacuated all the air from the apparatus, and then he adds the mixture of gases to the correct pressure. So we have hydrogen, methane, ammonia. Then the water in the flask was boiled, and the spark discharge was run continuously for 1 week. Now, at the end of the experiment, Miller was very careful to add 1 ml of saturated mercury chloride solution, and that was added to prevent the growth of any potential organisms. Stanley Miller was very conscious that the organics that he found in his mixture could have been just some trace contaminant, perhaps some fly might have gotten into the apparatus, and his colleagues were supposedly very vocal on the subject. So he was very careful when it came to sterilizing the mixture. Then the mixture obtained was separated from, I guess, the solid residues, and it was fractionated to some extent. The mixture was evaporated to remove volatile amines. He added sulfuric acid and then evaporated to remove the volatile acid constituents before neutralizing the extract with barium hydroxide, filtering it to remove any precipitated barium salts, and then concentrating it down. The concentrated neutralized extract was then chromatographed on paper. So this is a very simple analytical technique, and many of you might have done an experiment where you separated out the constituent pigments of ink on paper by just eluting the paper with some water. Here we can see perhaps a more elaborate version of that experiment with his extract. This is a 2-dimensional paper chromatogram. So the idea is that you would put a spot of the mixture in one corner of the paper, you would elute it with one solvent mixture, then you turn the paper by 90 degrees, and then you elute it with a different solvent mixture. So in this case we have a mixture of butanol and acetic acid, and in the opposite direction we have a mixture of phenol. Now this enables a separation of the different amino acids because each amino acid has a different affinity for the paper, and it also has a different affinity for the different solvents. So in this way he is able to resolve the mixture in 2 dimensions rather than if you were to use the same solvent twice, you would just see a diagonal line or a diagonal series of spots. Now he detected the amino acids using a reagent called ninhydrin, and although you can't see in this black and white photo, but ninhydrin would give different shades of purple spots unique to each amino acid. And he compared these with the spots formed by various analytical standards. So what we see is that he found aspartic acid, the very simplest amino acid, glycine, a slightly more complex amino acid in alpha-alanine, the isomer of alpha-alanine, which is beta-alanine, and alpha-aminobutyric acid, which is a non-natural amino acid. This might not look like more than a few splodges on a piece of paper, but this was a truly significant result, indeed a a groundbreaking result. He had established that amino acids can be formed through just sparking, let's say, lightning through an atmosphere and condensing them all in solution. So we can see that essential biomolecules, that is amino acids, which are the building blocks of proteins, can be formed by a purely natural abiotic mechanism. And so the field of prebiotic chemistry was spawned. Here is the same paper chromatogram with the chemical structures annotated on the slide. Now, I guess a lot of you are not chemists, so I'll just briefly explain the pictographical conventions we use when representing molecules in this way. You can see certain atoms are defined, so sometimes we can see a carbon atom and sometimes we can see a nitrogen atom or a hydrogen atom. But in many cases they're not. So the nodal points in these structures, for example, are carbon atoms, and hydrogen atoms are implicit in these structures, so they're generally not shown. So for example, to take the molecule glycine, we see we have the amino group on the end and we have the acidic carboxylate group, and in the middle we have a carbon bonded to two other hydrogen atoms. So there we have the simplest amino acid, and you can see why they are called amino acids, because they have both an amino group and an acid group in them. So nice and simple. Of course, Stanley Miller continued to work on spark discharge chemistry, and he made more elaborate experimental apparatuses, and he made far more rigorous analyses of the mixtures formed by spark discharge chemistry. So here is the second paper of his from his PhD, which was published in the Journal of the American Chemical Society. And we see that the experimental setup is more or less the same. He takes the mixtures of gases, ammonia, methane, water and hydrogen, and he subjects them to spark discharge over the course of a week. And he then fractionates the mixture of compounds in various ways. Now, in this paper, he actually describes three more evolved apparatuses. But here we can just see the first example, which is more or less the same as the original apparatus, except that the reactor headspace has been increased and the design of the electrodes for the spark discharge has been improved upon. Now, I said earlier that the mixture of compounds was fractionated. Well, in fact, they were very rigorously fractionated in this paper, and this work must have taken a substantial amount of time. To do this, they used ion exchange resin, so we see Dowex 50 here, which is just, it's a kind of ion exchange resin that you might find in showers, for example, to soften the water. I can tell you the water in Strasbourg is very hard, or you might use it to soften water for drinking purposes. In any case, the resins enable the separation of very acidic materials from very basic materials, because the materials will absorb first onto the resin and then they can be washed off in sequence. Now, he was able to identify, using this fractionation procedure, up to 25 new amino acids, so far more than in the original experiment, and he was also able to see that certain hydroxy acids were formed, and I will show you the chemical structures of some of these. Here we can see just a flowchart of the separation procedure, which is very detailed, and you don't really need to worry about the details. Suffice to say that he was able to resolve the mixture of amino acids into neutral and basic amino acids and the acidic amino acids just by using these various resins. He was also able to separate contaminants such as boric acid and silica, and these built up in the mixture over the time simply because the glass, which is a borosilicate setup, degrades. So he wanted to remove all of these artifactual products. Again, we see that he used two-dimensional paper chromatography to identify the mixture of amino acids formed. And on the next slide, I've annotated the chromatogram with the various chemical structures. We can see here the same types of amino acid that were formed in the original spark discharge experiment. We have glycine down here, we have aspartic acid, we have alanine, we have the alpha-aminobutyric acid, we have beta-alanine, and you can see that beta-alanine is a structural isomer of alpha-alanine, has the same composition, but the amino group is found in a different part of the molecule. So in this case it is 2 atoms removed from the acid functional group, and in the case of the alpha-alanine, which is the natural form, it is 1 carbon atom removed from the carboxylic acid functional group. We also see here sarcosine, which is N-methylglycine. This is a natural amino acid, and it's probably formed in this spark discharge experiment by a reaction of methylamine, which is produced by recombination of the gases in some sense. I'd just like to focus your attention on the last few sentences in the abstract, which are highlighted here, because we begin to see what kind of chemistry is taking place in the spark discharge apparatuses. He says that hydrogen cyanide and aldehydes are direct products of the discharge. And although there is insufficient evidence, the synthesis of hydroxy and amino acids may be through hydroxy and amino nitriles in solution. So let's look at what this chemistry means in real terms. And we'll begin by looking at the chemistry of hydroxynitriles, which are these type of intermediates, and hydroxy acids. This is a two-step process in which first hydrogen cyanide adds to the aldehyde to give this hydroxynitrile or cyanohydrin intermediate. We see that this intermediate has the carbon-nitrogen triple bond that emanates from the cyanide molecule. Carbon-nitrogen triple bonds react with water, that is to say they can be hydrated, and in fact it requires 2 equivalents of water, and we lose 1 equivalent of ammonia, and we form in the process the acid functional group. So that's the chemistry of alpha hydroxy acids. Now let's look at the slightly more complex example where we factor in the presence of ammonia and we form amino acids. In this case, ammonia first reacts with the aldehyde, and that gives us an imine, which is a molecule with a carbon-nitrogen double bond in place of a carbon-oxygen double bond. The imine is then trapped in an analogous fashion to before with the cyanide molecule to give an aminonitrile. And again, the aminonitrile is just as susceptible to hydrolysis as the hydroxynitrile we saw above. And we see that hydrolysis proceeds with, again, elimination of 1 equivalent of ammonia, and it results in the formation of our alpha amino acids. This lower piece of chemistry is a very classical piece of organic chemistry, and it's called the Streck reaction. It was discovered back in the 19th century. So it's a very well-established, well-known, well-characterized piece of chemistry. And this probably accounts for the formation of amino acids in the spark discharge chemistry. We saw at the end of the preceding section that the chemistry of hydrogen cyanide is central to the scheme, and it enables the synthesis of amino acids, the precursors to proteins, in a purely abiotic manner. Now let's take a look at the next fundamental piece of the jigsaw, and this was contributed by Juan Oro first. And this involves mapping the chemistry of hydrogen cyanide. So the next paper I'm going to talk about was published in 1961 in Archives of Biochemistry and Biophysics by Juan Oro and Kimball. And they made a seminal discovery. That is to say that hydrogen cyanide in solution on its own will spontaneously oligomerize, and it will give this molecule adenine. Now, adenine is a principal component of the nucleoside adenosine, which in turn is a principal component of our RNA, an information storing polymer. And adenine is a very stable pentamer of hydrogen cyanide, as I'll show you on the next slide. And the authors note that adenine is this essential building block of nucleic acids, And since it can be formed simply through the action of hydrogen cyanide, ammonia and water, which are natural constituents of the solar system, these experiments are considered of significance in relation to the problem of the origin of life. I think, again, we would all agree. What does this look like in schematic form? Well, we can draw this process very simply, and we can say that 5 molecules of hydrogen cyanide combine together to form 1 molecule of adenine. Sorry, this should be adenine, not adenosine. Adenosine is the nucleoside. This is a little tricky to see, and so as you would expect, this is far from a linear, simple pentamerization process. In fact, the mechanism is very complex. But I hope for now that you can see, if you follow the carbon-nitrogen units, I hope you can see 5 of them. You see one here, you see one here, you see one here, and one there. And I will show you in time how this process works. But for now, let's just leave the Oro result as stated on this slide, and we can leave it to Leslie Orgel to deduce the mechanism of this oligomerization process. Now, Leslie Orgel is quite a famous figure after Stanley Miller in prebiotic chemistry, and it's often joked that if you have an idea that Leslie Orgel didn't have, you're doing very well. Leslie Orgel did lots of very seminal work, but today I'm just going to talk about his study. Well, sorry, I'm actually going to talk about a few pieces of his work, but firstly, I'm going to talk about his study, which is mapping the oligomerization of hydrogen cyanide. He opens this paper by saying there is much evidence suggesting that hydrogen cyanide was important in prebiotic synthesis. We know that from the work of Oro, we know that from the work of Stanley Miller. And that in mildly alkaline solution, hydrogen cyanide is hydrolyzed to form imide and even formic acid, and these are molecules that we've seen in the protoplanetary disks. While polymerization of cyanide is apparently quadratic in hydrogen cyanide, so it's actually quite favorable a process. They see that within a certain pH range, which is relatively close to neutral, cyanide can be dimerized and thence oligomerized. And they also note that freezing out of the ice in the water permits very effective concentration of hydrogen cyanide. So this is what they refer to as eutectic phase oligomerization. So that's when you freeze out the ice. The organic molecules that are present in the water don't necessarily go into the ice, but they remain in solution, and they become very, very concentrated to the point that spontaneous oligomerization of these very concentrated molecules becomes a favorable process. Just moving on, they note that there are many very reactive intermediates in this oligomerization process, and I'll show you the structure of these. Particularly, we should note this very reactive trimer, which is called amino-malononitrile, and a relatively stable tetramer, which is called diamino-malonitrile. Again, I'll show you the structural formulae of these molecules later. They also started to look at the photochemical rearrangement of these molecules, and were in fact instrumental in highlighting the value of photochemistry in a prebiotic context. And we'll look at some examples of ultraviolet prebiotic photochemistry later on in the talk. Okay, let's look at some chemistry. This slide is quite complex, and I guess it's probably baffling to many of you, so let's cut it up into little pieces and we'll go through it step by step. Let's start at the top. Let's find our cyanide first. There it is. So we see that cyanide can react with water to form formamide, and it can also react with ammonia to form amidine. And these are reactive molecules in their own right that can undergo additional transformations, and we'll see later on in the scheme that they come back and participate in these relatively complex oligomerization processes. Before I go on to the next slide, I'd just like to point out the dimerization step. So we can see that cyanide can actually react with itself to provide a fairly reactive dimer, and this is called imino. Remember, an imine is a molecule with a carbon-nitrogen double bond. This is called iminoacetonitrile. Acetonitrile would be the molecule with a CH3 or methyl unit bonded to carbon-nitrogen triple bond. Okay, let's see how the complexity evolves. Well, cyanide can then react with the cyanide dimer to make this cyanide trimer. And this is indeed a very reactive molecule. It's very susceptible to hydrolysis. And in the absence of additional cyanide, water will add once and then twice to the triple bonds of the amino malononitrile intermediate to produce what is actually a very crystalline intermediate. I've done this chemistry in the lab, and yeah, I can confirm that this is an incredibly crystalline molecule. Probably the largest crystals I've ever grown in the lab were of this molecule, but it's a synthetic dead end in many ways. It doesn't really get us very far. I mean, perhaps we can hydrolyze it to glycine, but that's not terribly exciting chemistry. What we're more interested in the context of building up in complexity and continuing with our oligomerization scheme, is the addition of a fourth molecule of hydrogen cyanide to the trimer. And then we form this mixture of stable but fairly reactive cyanide tetramers. We see on the right this molecule diamino-malanitrile, and on the right-hand side we see diamino-fumaronitrile. The names of these molecules actually derive from the natural acids, which are maleic acid and also fumaric acid. And these are biosynthetic intermediates in various metabolic cycles. Note the photochemical interconversion of these molecules, and this is fairly standard for molecules that contain carbon-carbon double bonds. The carbon-carbon double bond can be excited and indeed broken by UV photons. And so the stereochemistry, that is to say the relative orientation of the groups around the double bond, can be scrambled. If we then add one molecule of formamidine to the diamino-fumaronitrile cyanide tetramer, we get to this rather interesting looking imidazole. And imidazole is just a fancy term for this cyclic structure here. And we can even add a second molecule of formamidine to close up a ring in this position, and then we get to the nucleobases adenine and guanine. The nucleobases listed here are xanthine and hypoxanthine, and these are just hydrolysis products of the various nucleobases that we would form by reaction with cyanide and its oligomers. So just to recap, we saw this very simple scheme from Juan Oro's paper, but we now know that the route that cyanide takes to get here is a really circuitous one. But because of the thermodynamic stability of adenosine, there is such a driving force, adenine, there is such a driving force for its production that we're able to form substantial amounts of adenine from hydrogen cyanide. So far, we've looked at the formation of, let's say, relatively simple molecules, even if the mechanism by which these molecules formed was relatively complex. We've seen the amino acids of Stanley Miller's spark discharge chemistry, and we've seen the nucleobases of Juan Oro and Leslie Orgel studies based on the oligomerization of cyanide. Now, let's look at some more complex chemistry, and the example I've chosen to show you today is Leslie Orgel's early synthesis of the pyrimidine nucleosides, and I will explain what all of these terms mean in structures later on. I suppose I should just say a few more things about the significance of nucleosides at this stage, and that is that just as amino acids are the building blocks of biochemical proteins, nucleosides are the building blocks of the oligomers that we know as ribose nucleic acid and deoxyribose nucleic acid. These are the information storing polymers. that contain the chemical blueprints for everything a cell might need to make, and indeed the chemical blueprints of life. Orgel's findings are very nicely laid out in this paper, but let's look at the abstract first. And we see that ribose phosphate, or ribose, these are just sugars or sugar phosphates, react with cyanamide in aqueous solutions to first provide an amino-oxazoline derivative, and I'll show you what this means in a later slide. Moving on, we find that these oxazoline intermediates react very nicely with cyanoacetylene, and recall cyanoacetylene is a relatively simple molecule that again we find in protoplanetary disks. We also find it in cometary ices. I think cyanoacetylene has even been observed in the atmosphere of Titan, along with several more complex variants of cyanoacetylene where there are additional acetylene units all in a linear arrangement. And so the reaction of cyanoacetylene with the amino-oxazoline gives us a precursor to a nucleoside, which is then opened up with water to reveal the nucleoside. They then found a very interesting reaction that is of true significance, and that is this anemorization and epimerization reaction. Now again, anemorization is just a fancy chemical term for a kind of flipping process, which is very important when it comes to the spatial orientation of the nucleobase part of the nucleoside molecule. I will show you. And of course they discuss the prebiotic relevance of these processes. Let's take a look at the chemistry now. If we start in the bottom left-hand portion of the slide, we can see ribose, that would be the sugar molecule without the phosphate, and they've included the phosphate in brackets because the chemistry works equally well if there's a phosphate group on the terminal hydroxyl group of this sugar molecule. Cyanamide reacts very rapidly, in fact, with sugars because sugars are aldehydes in disguise. And you can imagine that cyanamide is an amine, and amines react with aldehydes to give imines from before. The reaction gives this structure where the sugar part of the molecule has been abbreviated with these dotted lines, but the interesting part of the molecule is this heterocyclic structure here, and we call this an amino-oxazoline. When it comes to the reaction of ribose with cyanamide, the resulting amino-oxazoline is a very crystalline, very stable molecule. And we suspect that these molecules are key intermediates when it comes to the origin of ribonucleotides, and that simply relates to their crystalline stability. So you could imagine that they would form in a pool on the primordial Earth, and that they would crystallize out and in that way protect themselves from degradation in the otherwise harsh environment. We see that this amino-oxazoline then reacts with cyanoacetylene, and it reacts by this nitrogen atom forming a bond to the terminal carbon atom of the cyanoacetylene molecule. This is a very reactive intermediate that then spontaneously cyclizes to give this six-membered ring structure here. This structure in turn is relatively unstable. It can be isolated, but it reacts quite rapidly with water to cleave this bond here, and this releases what we would call the nucleobase, and we generate the sugar again here. Now this molecule, if we ignore the phosphate for a moment and we just focus on the conjugate between the sugar and the nucleobase, this is what we would call a nucleoside. So remember that going forward. A nucleoside is a conjugate between a sugar molecule and a nucleobase. This is actually the non-natural form of the nucleoside cytidine. And you remember I said that anemorization is a fancy word for a flipping process. We have to imagine this molecule, how it looks in 3 dimensions. So imagine that this sugar molecule forms a kind of plane that projects out of the board. And then imagine that the nucleobase portion of the molecule projects down. So this is the unnatural form. The natural form of the molecule is the one in which the sugar projects out and the nucleobase projects up. And that's very important for its base pairing properties. if the nucleobase did not project up, you would not be able to form these beautiful double helices that we associate with the nucleic acids RNA and DNA. So we see here just a table showing the various results, and the main result is here that if we take this unnatural alpha cytidine isomer and irradiate it with UV light of 253 nanometers, I think, wavelength, then we are able to at least partially flip the configuration or the orientation of the nucleobase portion of the molecule to the natural form of the molecule, which is this beta cytidine. And it's very interesting that you can actually start with sugars other than ribose. For example, you could start with the sugar arabinose, which is a structural isomer of ribose. And you could also form a small quantity of the natural beta cytidine nucleoside. So this is quite remarkable chemistry. This is a slide which shows, again in pictographic form, what I was trying to illustrate with my arms. So we see here a convention used where a dotted line means that the bond projects down, or in this case it actually projects into the board. And we see a bold line, which means that the substituent projects up, or in this case, out of the board. So again, you have to imagine these molecules and how they look in 3 dimensions, and we're just interested in this equilibrium process whereby you can flip the molecule from one side of the sugar plane to the other side of the sugar plane simply by using ultraviolet photons. For the chemists in the audience, you might want to ponder the mechanism by which this transformation takes place, and that is that it involves homolysis of the carbon-oxygen double bond, so we form a diradical intermediate, and the oxygen radical is probably able to abstract the hydrogen atom from this position. The intermediate is a structurally planar one, where the bond connecting the sugar to the nucleobase is capable of rotating, and so the hydrogen atom can be transferred intramolecularly via this oxygen atom from, in this case, the top side of the sugar to, in this case, the bottom side of the sugar. So truly remarkable but very simple chemistry. It requires nothing but UV photons. I think that's pretty cool. The other important observation that Leslie Orgel made in this work, and this is an observation that didn't make it into the abstract, so you can thank me that I'm highlighting it for you, is that hydrolysis of cytidine or cytidyllic acid, this is the nucleotide, this is the molecule with the phosphate group attached, is a spontaneous process, and water will add to cytidine to give over time some uridine. We can see as either the unnatural or the natural form of cytidine is just incubated in water over the course of several weeks. It is consumed, and we gradually build up uridine, which is another nucleoside in this chemistry. So this really means that we get 2 nucleosides for the price of 1, simply by spontaneous hydrolysis of the molecule. Again, this result is significant because it increases the number of nucleosides that could have been available on the early Earth. And of course, we need a variety of nucleosides to form a genetic code. In each case, we need 4 ribonucleic acids and we need 4 deoxyribonucleic acids. So the more we can get from any one process, the better, because the closer we are to forming an information storing molecule. Again, the chemistry just looks like this. We see here we have a nitrogen-carbon single bond, but actually this molecule is dynamic in a sense because we can push electrons from this nitrogen atom into this ring structure and move the electrons to this nitrogen atom. And in doing so, we could create a carbon-nitrogen double bond, which is an imine that we saw before. Imines are susceptible to hydrolysis and water can add to them. And so the water molecule, the oxygen atom from the water molecule, becomes incorporated into this ring structure, and we eliminate one molecule of ammonia. And the conditions required for this transformation are very mild indeed. In fact, in the lab they just used a little sodium bicarbonate in water at pH 8, so very, very mild conditions. Sanchez and Orgel give a refreshingly honest account of their results. They say the reactions they have described lead to the formation of the derivatives alpha and beta, arabinocytosine in this case, but not to the derivatives of the important nucleic acid component, beta-cytidine. However, the photochemical isomerization of the alpha-cytidine leads to the beta, but they concede the yields are relatively low. So, although they have described a potentially prebiotic route to cytidine and uridine, they concede that it is not very efficient. But it was, at the time, and still remains, a very plausible prebiotic route to a natural nucleoside. And as I said before, we actually get two natural nucleosides out of this. Now, I said at the start of the talk that many questions remain unsolved in prebiotic chemistry. The synthesis of nucleosides and also nucleotides is perhaps an exception in that we have made a lot of progress in the last 20 or so years in this area. And for those who are interested, I would recommend you check out the work of particularly John Sutherland and Matthew Powner, who have essentially taken all this chemistry and serially improved upon it through the use of molecules like hydrogen sulfide to produce sulfur-containing intermediates, which are far more reactive than the oxygen-containing intermediates that I showed you. And these subtle variations enable the chemistry to become far more efficient. The yields become a lot higher. The photochemical epimerization, for example, is far more efficient. And so potentially we will be able to produce far more of any given nucleoside than Orgel dreamed of. Now, in the previous section, we saw how ribose can be used in the synthesis of nucleosides. We also saw that instead of ribose, we could use ribose phosphate. So that is a sugar molecule with a phosphate group already attached. The phosphate group, it turns out, is very important because we need the phosphate to connect all of the sugar molecules into our ribonucleic acid, or indeed deoxyribonucleic acid polymers. That's to say, without the phosphate there, we cannot polymerize these molecules. So a fundamental question is, how do we add phosphate to nucleosides to produce nucleotides, which can then be polymerized into oligonucleotides or indeed genes? So that's what they are. And so this is again something that Leslie Orgel and his group weighed in upon. And in 1971, they published a science paper where they elucidated a very simple means by which phosphate can be added to nucleosides in very high yields. And the way they do this is simply by drying out a mixture of urea and ammonium chloride with the respective nucleoside and heating the dry mixture in the temperature range 65 to 100 degrees. And they don't have to use inorganic phosphate on its own. It's perhaps unlikely that inorganic phosphate would have been freely available on the early Earth, but it is quite conceivable that several phosphate-containing minerals could have substituted for inorganic phosphate, and the authors found that the mineral hydroxyl apatite is capable of performing this chemistry. So here is just a table summarizing their various results, and they used the nucleoside uridine, which I showed in the previous section, and over the course of 24 hours, all we really have to look at is that the incorporation of inorganic phosphate is incredibly efficient. Now, the authors used these abbreviations or acronyms for their phosphorylated derivatives, and I will attempt to show you what this is. Essentially, they just put a P in front of any molecule that has a phosphate attached to it, and the position of the P in the acronym tells you whereabouts it is. The exclamation mark just means that the phosphate is now connected to two hydroxyl groups, and that's a cyclic structure, which is a special kind of activated phosphate intermediate. Here we see just in graphical form, we see nicely the consumption of uridine, and we see the production of various phosphorylated uridine derivatives. We don't have to worry particularly what they are, but we see that the total incorporation of phosphate is remarkably efficient. What do these molecules look like then? Well, we take our uridine, which has 3 available hydroxyl groups, and hydroxyl group is simply an OH group here. We treat it with urea, inorganic phosphate, which is abbreviated as PI here, and ammonium chloride. And we heat it in the dry state for however long we want. 24 hours is obviously good enough. And we see that the phosphate molecule can be added to every single possible hydroxyl group within the molecule. So we see over time all possible combinations of phosphorylated products formed in this chemistry. Fortunately for us, this phosphorylation reaction is not specific to uridine. It's actually a very general one, and phosphate can be readily incorporated into all of the different nucleosides. So we have cytidine, which is a purine, structurally analogous to uridine, and we have the cytidine is pyrimidine, and we have the purine nucleosides adenosine and guanosine. The reaction is not quite as efficient for adenosine and guanosine. In the case of adenosine, that's probably because we start to observe some partial degradation of the molecule. It turns out that the purines are not as stable as the pyrimidines. And in the case of guanosine, it may be a result of the poor solubility of guanosine. So when the mixture is drying out, the guanosine might precipitate to the point that it doesn't get to mix very efficiently with the urea and ammonium chloride. So when the solid is heated, it's not able to meet the other reactants. Another interesting feature of this chemistry is what's called transphosphorylation. So once you have a phosphate attached to the nucleoside, it can actually move around. So it might be on the 5' hydroxyl to start with, but if you treat it with urea and ammonium chloride, we see that the phosphate can be migrated. And in some cases that might be advantageous. For example, we might be able to convert the 5' phosphate of a given nucleoside to the 2', 3' cyclic phosphate, which is an activated intermediate and can spontaneously oligomerize. So just to illustrate this in graphical form, we see here an example where we have the 5' phosphate of uridine. We treat it with urea and ammonium chloride and we heat it. And in some cases the phosphate just comes off. But in others we see that it is taken off and then it is reattached in different parts of the molecule. And again, we see that all possible combinations of products can be formed in this chemistry. So potentially, each product could be used in a different type of prebiotic transformation, some of which we have knowledge of, some of which undoubtedly remain to be found. And this is the same chemistry. We don't have to start with the 5' phosphate, we can start with the 3' phosphate, but the chemistry proceeds in exactly the same way. Now, to this point, we've looked at amino acids, the building blocks of proteins. We've looked at nucleosides, and we've looked at how you add a phosphate molecule to a nucleoside to make a nucleotide. And nucleotides are the building blocks of our information storing polymers like RNA and DNA. But there is another class of molecule that is vital to life on Earth, and these are the lipids. To make an analogy to emphasize the importance of lipids, if we were to consider life as an egg, well, the lipids would be the shell. And what is an egg without its shell, outside of breakfast time? Now, I haven't actually shown the chemical structure of a lipid in the following slides, but just to explain briefly, a lipid in general is a molecule that consists of two parts. It consists of a long carbon chain and a polar head group. So the polar head group could be a phosphate, it could be an oxygen atom, something like that. The polar head group interacts very strongly with the water, so we call that hydrophilic. And the long carbon chain is a very greasy kind of molecule, and that is very hydrophobic. It doesn't like the water at all. So the defining feature of a lipid is that if you were to throw a lipid into a body of water, it will spontaneously arrange itself with other lipid molecules such that the polar head molecule is oriented into the water and the greasy carbon-rich tail of the molecule finds another carbon-rich tail so that it can escape the water altogether, and it will actually form a bilayer structure characteristic of a natural membrane. Lastly, I would just say that there is enormous diversity of lipids in nature, and so lipids are unique in that sense. We have a relatively small number of molecules that make up our protein, 20 to 22 natural amino acids, plus or minus some variants. And our genetic alphabet is even more restricted. We have only 4 components that make up the RNA alphabet, and we have another 4 components that make up the DNA alphabet. In contrast, it's been estimated that there are over 500 different classes of lipids, which is staggering, and it is unclear why. But of course, the question we have to address is, how could lipids have come into being on the early Earth? We know that lipids are found in carbonaceous chondrites, these carbon-rich meteorites that plough into Earth from time to time. And Juan Oro, back in the 1970s, started pondering how they could form on meteorites, and analogously, how could lipids form on the early Earth from a relatively small pool of ingredients? Could the chemistry that takes place on meteorites be mimicked on the early Earth? And he looked at a kind of chemistry which is called Fischer-Tropsch chemistry. Fischer-Tropsch chemistry was developed actually in the 19th century and was really pushed in the 20th century in coal-rich yet oil-poor countries. So Fischer-Tropsch chemistry enables us to transform a mixture of gases, principally carbon monoxide and hydrogen, in the presence of a heterogeneous catalyst. And in fact, only 4 metals are able to catalyze this process. We have iron, nickel, cobalt, and ruthenium. And the gases adsorb onto the surface of the metal, where they are reduced by the hydrogen, which is probably also adsorbed onto the surface. And 1 carbon atom at a time are connected on the surface of the metal. So we see how by sequentially adding carbon atoms, we can form a nice long carbon chain like we find in the lipids. So Juan Oro decided to look at Fischer-Tropsch chemistry and to see whether he could take the gases deuterium and carbon monoxide in the presence of meteoritic iron. In this case, he used metal from a meteorite that landed in Canyon Diablo in the US, and heated the heterogeneous mixture to between 195 and 370 degrees Celsius for a range of times. Their analysis showed that they were able to produce alkenes and alkanes and also alkenes, but most importantly, they were able to produce fatty acids, and fatty acids are the very simplest lipids, the very simplest membrane-forming molecules. So how did he experimentally evaluate this possibility? Well, he took some meteoritic iron from the various samples, or indeed the Canyon Diablo or Orgueil meteorites, and he adds potassium carbonate to it. Now, potassium carbonate might seem like an unnatural additive, but I think it was just added to make the chemistry go a little better. It won't make the chemistry, so the chemistry is spontaneous, but the presence of potassium carbonate will just increase the efficiency of the chemistry. So really it has been added to help out in the analytical steps, which is relatively complex for these mixtures. They add the mixture of gases to the reactor, and they seal the reactor, and then they heat it in a furnace. Now at the end of the experimental period, they cool it down very carefully and they evacuate all the gases, and then they wash the inside of the reactor with benzene and methanol, because it was the 1970s. People don't use benzene so much in the lab these days because it's a nasty carcinogen, but benzene is actually a very practical solvent to use. It's capable of dissolving a very broad range of analytes, so that's probably why they used it. And they take their extract and they condense it down under a flow of nitrogen, and then they analyze their condensed extracts by gas chromatography. Now, gas chromatography is a means of analyzing, let's say, relatively volatile molecules, and the molecules in the sample can be separated on a column based on their different vaporization temperatures. So we see here an analysis of the alkane fraction, and we see that there would be an awful lot of short chain alkanes, things like propane, butane, things like that, pentane and so on. But really what we're interested in terms of our lipids are these longer molecules. Now we see two interesting features in these experiments. One is that at high temperature we don't see very many long chain alkanes formed, and that's because the high temperature causes the alkanes to crack, so they fragment, and that's why we see predominantly shorter chain products. However, if we lower the temperature to 195 degrees, we then see this peak between the C16 and C18 regions in the distribution. And this is quite intriguing in a sense, because the natural lipids that we find in our membranes contain generally between 16 and 18 carbon atoms. There are, of course, many exceptions, but the C16 fatty acid is palmitic acid. You might have seen palmitic acid soaps in your various soaps you buy in the supermarket. And the C18 fatty acid is called stearic acid, and again, that's a prominent constituent in a lot of membranes and a lot of commercial products that derive from the breakdown of membranes like soap. From an origins of life perspective, the most significant result, however, is the production of long-chain fatty acids. And again, we can see here just these little blips in the baseline that correspond to the long-chain fatty acids, and we see that we're able to make up to 16 carbon chain length fatty acids just by this very primitive Fischer-Tropsch process. Now, as I mentioned at the end of the section where I dealt with the synthesis of nucleosides, a lot of subsequent work has been done on Fischer-Tropsch chemistry in a prebiotic chemistry context. And for those of you who are interested, I would recommend you search out the works of Tom McCollum and also Bernd Simonite, who have looked at Fischer-Tropsch chemistry in hydrothermal systems. I would add that although the system is indeed a hydrothermal one, the reaction is still probably a gas phase reaction. And so the reactions actually probably take place in the headspace of the reactor and are catalyzed on the walls rather than in aqueous solution. I'm just going to finish with a section that deals with the synthesis of phospholipids. Phospholipids are more complex lipids, and indeed they are the types of lipids that we find in the vast majority of membranes in all kinds of living organisms on Earth. We saw in the previous section how long-chain fatty acids could be produced by Fischer-Tropsch or Fischer-Tropsch-like processes on the early Earth. But it turns out that fatty acids, although they are capable of forming membranes on their own, do not form particularly stable membranes, and that's because they are susceptible to precipitation in the presence of divalent metal cations. For example, magnesium in water will actively precipitate fatty acids, and it will form a kind of soap scum on the surface of a liquid, of water, sorry. So David Diemer and his colleagues pondered whether it would be possible to form more complex lipids which are more stable to the divalent metal cations that you find in solution, and the example he wanted to look at was that of phospholipids, specifically glycerophospholipids. So he opens with some statements that I guess we can all agree upon. It's generally agreed that stable membranes were prerequisite to the assembly of the earlier self-replicating system. So think back to the eggshell analogy. What is an egg without its shell? If we cannot contain the information coding polymers, if we cannot contain the catalytic or structural peptides that form on the primitive Earth, then we're not in a position to make any progress towards something that might resemble a living organism. We have to contain them. Phospholipids are, of course, ubiquitous in modern biology, and they form extremely stable lipid bilayers, which is fortunate for all living things on Earth. They were interested in figuring out how to combine the various ingredients to make glycerophospholipids. So in this case, we have to combine somehow 3 ingredients. We have to take our long-chain fatty acid, which is the hydrophobic part of the lipid. We have to combine a glycerol molecule, which is an alcohol with 3 hydroxyl groups, and we have to attach a phosphate group somehow to this conglomerate of a molecule. And we've seen Leslie Orgel's phosphorylation method before. Interestingly, they suggest that the major source of energy for this process was probably the sun, not ultraviolet light. Now this makes a lot of sense in the context of lipids, because lipids are relatively transparent to ultraviolet lights, and so they don't interact at all. And if we want to condense molecules, we need some way of heating them and eliminating water from the system. They were also interested as to whether clays or minerals such as silica could catalyze dehydration processes. Silicates are very abundant materials in nature. A lot of our rocks are magnesium aluminosilicates, so we can use silicates to promote any kind of chemistry without worrying too much about their provenance. Clays, on the other hand, may have been relatively rare on the early Earth, but it is the structure of the clays that we are most interested in. Clays form sort of layered structures which contain lots of holes, and we can imagine that the various organic molecules diffuse into these holes, and in tiny acidic cavities within the clay structure, potentially dehydration reactions can be facilitated. Hargreaves, Mulvihill and Diemer make 3 key observations in this paper, and the first is that heating to dryness mixtures of glycerol, fatty acids, or aldehydes or alcohols gives a range of different glycerolipids. Now remember back to the work of Stanley Miller. He used paper chromatography to separate out his mixtures of compounds. Well, here we see an autoradiogram, which is generated from a silica gel thin layer chromatogram of mixtures taken from reactions containing glycerol and either fatty acids or fatty aldehydes. So in this case, he uses a carbon-14 labeled glycerol, and so we can follow the progress of the reaction just by separating out all of these molecules on a plate using different solvents. And because they are radioactive, we can detect them on photographic film. So we see here, if we take glycerol with carboxylic acid or a carboxaldehyde, we can form a great variety of products that derive from condensation of these 2 classes of molecules together. The reactions are more efficient if he uses the clay kaolin, or if he uses silica as a proxy of an aluminosilicate containing mineral or rock. We see that the reactions involving carboxylic acids and indeed carboxaldehydes are far more efficient than those containing alcohols, And that is because it's very difficult to dehydrate an alcohol to form an ether. It's exceptionally difficult, in fact. But nevertheless, we're able to produce these types of molecules. DGs are diacyl glycerols, and 2,3 diacyl glycerols are in fact prominent biosynthetic intermediates in the synthesis of our phospholipids and the phospholipids of every other organism. The preparative yields, which are quantified by scintillation counting based on the radioactive products, are pretty low. We're looking in the region of less than 10%. However, we're able to form a diversity of products, and in fact, not all of these products were assigned structures. We don't know exactly what they are. Of course, it's very difficult to analyze when we don't have very much material, and I guess at the time they were more interested in the fact that they could produce defined lipids rather than analyzing everything ad infinitum to characterize the mixtures fully. I said that not every product was characterized, but perhaps most interestingly, in the addition to ester-containing molecules, and these are the 1,3 and 2,3 diglycerides, we also see molecules that derive from condensation of alcohols, and these are probably ether lipids. And ether lipids are found in one particular type of organism on Earth, and these are the archaea. Archaea produce some phenomenally different ether lipids, and ether lipids are remarkably stable to the point that they can survive in rocks where archaea once lived for billions of years. And in fact, even I have analyzed some billion-year-old ether lipids. The second observation is that heating to dryness mixtures of these ether lipids with phosphate and dicyanamide. So in this case, dicyanamide is used as a condensing agent, just as Leslie Orgel used urea to condense phosphate with nucleosides. David Diemer uses dicyanamide to condense phosphate with the glycerolipids that were produced in the previous step. But again, unfortunately, the yields are pretty low. So we see here that taking a relatively complex mixture of lipids and phosphate and dicyanamide and heating it, they are only able to produce traces, or perhaps up to 0.2% yield of phospholipid products. Nevertheless, let's assume that ether phospholipids were present in the mixture, and that they probably derive from this molecule, which is chimal alcohol, which has a C16 carbon chain. Remember back to the Fischer-Tropsch, where I said that C16 lipids are quite prevalent in nature. And it's also possible that in this mixture we have formed some mixed species where you have one ether bond and one ester bond, but we really aren't sure. In any case, they were able to take their mixture of phospholipids and precipitate what little material there was, and they put this material into water and found that the phospholipids that were produced formed stable vesicles. Now, vesicles are sometimes also called liposomes. These are just spherical structures that are made up of lipids that spontaneously assemble themselves into bilayer membranes. And they characterized these vesicles by various kinds of microscopy. Now, I'm sorry the images aren't very good, but what we're really looking at are these dark splodges. These are the the vesicles as shown by phase contrast microscopy. Fortunately, they used some different techniques, and this is one called negative staining microscopy, in which they use uranyl acetate to stain these spherical structures, and it's very clear from this micrograph that we can see membranous structures in solution surrounded by bulk water. The last method they used to characterize these was quite an interesting method called freeze fracture electron microscopy. And this is where they take their mixture of vesicles in solution and they flash freeze it. They then cut the block of ice and then they imprint the ice onto a carbon film and then analyze the imprint microscopically, by microscopy, sorry. And what we're looking at are these spherical little bubbles which are formed from the lipids. That's all I have for you today. I chose to end this talk with something that at least looks like a cell. We have a ball of lipids, even though there's nothing inside. We can see how we might arrive at lipids, and we've also seen how we might arrive at the building blocks of every other class of biomolecule that would be vital for the early evolution of life. So, thank you very much for your kind attention.